hypertrophy that is defined as an increase in the cell volume and consequently an increase in the tissue volume. Hypertrophy may be due to a number of stimuli. Common examples are hypertrophy from loading on the striated skeletal muscle, which occurs, for example, with training, and hypertrophy from hemodynamic overload, which is caused by a chronic hemodynamic overload and leads to a cardiac muscle hypertrophy. Another example is that of the hypertrophy of the uterus during pregnancy due to the action of the estrogens binding to receptors on the smooth muscle cells. The hypertrophy from hemodynamic overload occurs with hypertension and valvular heart diseases, two pathologies causing the heart to beat at a higher blood pressure. The myocardium is normally between 1 to 1.5 centimeters thick, but when hypertrophic, it is over at least 2 centimeters. From a histological point of view, triphenyltetrazolium coloration indicates how much of the myocardium is still working correctly. But hypertrophy can also be biologically determined by a number of external stimuli, among which mechanical stretch. This is the case of the smooth muscle cells since there are some receptors perceiving the stretching membrane. Hormones and vasoactive peptides do represent another stimulus inducing hypertrophy. Another stimulus is then given by growth peptides, about which we're going to talk later on. These stimuli affect transaction pathways. While hypertrophy from G protein-coupled receptors is more common for pathological hypertrophy, the physiological forms of hypertrophy mainly involve the phosphatidylinositol 3 kinase and AKT pathway. These transaction pathways lead to the activation of some transcription factors that are involved in the process. Among these factors, we can find c-MYC and c-phos-c-Jun dimer, also known as AP1. A common effect of hypertrophy is embryonic gene expression, since hypertrophic cells are not only increasing in volume, but also changing their protein synthesis pattern. For example, gene re-expression may occur for beta-myosin, replacing alpha-myosin in the heart. Beta-myosin gene is an embryonic gene allowing a more effective contraction. To reduce the hemodynamic overload, the atrial natriuretic factor is instead produced by the ventricles as well. Moreover, an overall increase in protein synthesis occurs. The simplest example is that of hypertrophy from striated muscle training, in which a great amount of sarcomere proteins are synthesized. Another effect is that of the tissue that, by becoming hypertrophic, can start producing its own growth factors. This phenomenon, also known as amplification, is a positive feedback. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.